At first glance, this volleyball game has nothing to do with science. And yet there are surprising parallels between this ball and the research Sara Wickström and her team are conducting. She's interested in cells, especially in the effect that physical forces have on our body cells.
The same laws of physics that govern the shape of the volleyball govern the shape of our cells. So we have some sort of internal pressure that is resisted by a shell, which in the case of the volleyball is leather. It's more complicated in our cells, but essentially it's the same thing.
Volleyballs have to withstand a lot, just like our body cells, which are stretched, compressed, shoved, and pulled.
We are very interested in understanding how actually the cells will react to these forces. So if a disease alters the mechanical properties, suddenly these cells are actually not prepared for these forces, and this means that they have to react and change their structure, but sometimes this change is also bad for their everyday function, and that might disrupt their normal function.
How exactly does a cell cope with these challenges? Sara Wickström has found remarkable answers to this question, and she's developed groundbreaking experiments. This cell culture, for example, consists of endothelial cells that line our blood vessels. The sample is exposed to flow and shear forces like those acting in our circulatory system. This allows the team to observe how the cells respond to blood flowing past them in controlled conditions. A special pump system pushes this liquid across the cell layer as a blood substitute. When pressure and flow patterns in the vessels change, inflammation and vascular obstructions can occur.
What we see is the cells actually reacting and aligning in the direction of force, which is their usual reaction, but we are particularly interested in what happens in terms of molecules, how molecules and the cellular processes change in response to this shear.
Like our cells, a volleyball is exposed to the forces acting on it, but it cannot itself respond to them, unlike our body cells. The molecules inside our cells are able to actively process these forces. This is Sara Wickström's field of research.
What continues to fascinate me is cellular dynamics, how cells are able to react, to change dynamically their state, and to work together to form functional tissues and organs.
Deformations are converted into chemical signals inside the cell, an interaction of mechanics and gene activity that Sara Wickström can demonstrate directly within living tissue. It's a milestone in so-called mechanobiology and the basis for a remarkable discovery.
We have the surprising finding that the cells actually react extremely fast. Within seconds and minutes, actually they reprogram their cellular programs, and that is something that we are following up right now.
Another discovery is driving her research towards a better understanding of diseases.
Scan here, basically where the dermis is. Okay, scan is working.
The mechanical properties of tissues are precisely analysed and visualised here. Diseases such as cancer or fibrosis, in which scar tissues form, have long-term effects on cells and their function. These changes also directly affect gene regulation, which determines which genes are activated, when and how.
This should give the cells the ability to store the information of forces for a longer period of time. And that would be possibly important for diseases where tissues have experienced damage, and the cells might remember this damaged state, and that might impact their future behavior.
The team now want to find out whether cells have some kind of memory, which would allow them to revert back into an undamaged state. Mechanobiology also offers possibilities for the detection of diseases such as cancer. Here, a tumour subjects the cells to pressure. On the left, cancer tissue in a classical image. On the right, pressure-sensitive signalling molecules serve as cancer markers, the optical fingerprint of the tumour. Analysing a patient's tissue in this way helps to better determine the stage of their cancer.
I don't want to make concrete predictions, but of course, with maybe earlier diagnostics based on markers that we can develop or some sort of other diagnostics, I think we might be able to tackle diseases much earlier.
These days are yet to come, but Wickström's findings on how forces impact cell tissues could eventually lead to life-saving medical applications. Basic research with clear perspectives.
My goal is to make fundamental discoveries in biology that are also relevant for human health, and at the same time to train the next generation of scientists.